Hey everyone! Welcome back to channel. In today's video, we're exploring a crucial topic, quality control tests for tablets. These tests ensure that the medicines we take are safe, effective, and meet the highest standards of quality. Let's jump right in. Overview of tablet quality control. Tablets are one of the most common forms of medication, but before they make it to the pharmacy shelves, they go through rigorous quality control tests. These tests are designed to ensure that every batch of tablets meet stringent guidelines for safety, efficacy, and consistency. Why is this so important? Well, if the quality of the tablets isn't properly controlled, it could affect how the drug works in your body. Imagine receiving a tablet that doesn't dissolve properly or has inconsistent dosage. Scary, right? Let's look at the key tests that are performed during quality control. Key quality control tests. There are several essential tests performed on tablets to ensure they meet quality standards. These include appearance testing, average weight, hardness, thickness, friability, disintegration, dissolution, and assay testing. Let's break these down one by one. Test 1, Appearance Test. The first thing to check is appearance. The tablets should be uniform in color, size, and shape, without any cracks, chips, or discoloration. Even though this seems simple, it's important because any physical defects can indicate problems with the manufacturing process. Test 2, Average Weight. The average weight of a tablet is monitored during the production process to ensure uniformity. According to international pharmacopoeias like the United States Pharmacopoeia, USP, and European Pharmacopoeia, EP, tablets must meet specific weight variation limits based on their average weight. If a tablet weighs too little or too much, it can indicate issues in the manufacturing process. Formula for average weight. To calculate the average weight of tablets, the following formula is used, where sigma W is the total weight of the tablets in the sample, N is the number of tablets in the sample. Steps to calculate average tablet weight. Sample selection. Select a sample of tablets from a production batch. Typically, 20 tablets are chosen, but this number may vary based on the manufacturer's quality control protocols. Weigh the tablets individually. Each tablet is weighed individually using a precise balance to ensure accurate results. Sum of individual weights. Add up the individual weights of all the tablets in the sample to get the total weight. Calculate average weight. Divide the total weight of the sample by the number of tablets. This gives you the average weight of one tablet. Example calculation. Let's say you weigh 10 tablets, and the weights in milligrams are as follows: 501 milligram, 498 milligram, 502 milligram, 500 milligram, 499 milligram, 500 milligram, 503 milligram, 497 milligram, 501 milligram, 499 milligram. Sum of weights, number of tablets, N equals 10, N equals 10, N equals 10, average weight equals 5000 divided 10 is equal to 500 milligram. Weight variation limits. The allowable variation in tablet weight depends on the tablet's average weight, and this is governed by pharmacopoeia guidelines. For example, tablets weighing 130 milligrams or less, plus or minus 10% of the average weight is allowed. Tablets weighing more than 130 milligrams but less than 324 milligrams, plus or minus 7.5% of the average weight is allowed. Tablets weighing 324 milligrams or more, plus or minus 5% of the average weight is allowed. If the average weight of the tablets is 500 milligrams, for instance, the acceptable variation would be plus or minus 5%, meaning individual tablet weights should fall between 475 milligrams and 525 milligrams. Test 3, Hardness Test. The hardness test is a critical quality control measure in pharmaceutical manufacturing that ensures tablets have the appropriate strength to withstand handling during packaging, transportation, and storage without breaking or chipping. At the same time, the tablet must be able to disintegrate and release the active pharmaceutical ingredient, API, within the body as required. Tablet hardness testing is primarily focused on determining the mechanical strength of the tablet by measuring how much force is needed to break it under pressure. A tablet that is too hard may not disintegrate properly in the digestive system, while one that is too soft may crumble during handling. The balance of these properties is crucial for the tablet's performance and efficacy. Principle of the hardness test. In the hardness test, a tablet is subjected to pressure between 2 points, and the force required to break it is measured. This force is usually expressed in kilogram force or Newton, where 1 kgf equals 9.8 N. The tablet is placed between 2 anvils, and force is gradually applied until the tablet fractures. The hardness, or breaking force, is then recorded. Formula for tablet hardness. The hardness value is typically read directly from the hardness tester, but if you're calculating manually or converting between units, you can use the following relationship. Hardness is commonly measured in kilogram force, and high-precision instruments provide the force required to break the tablet. Standard hardness range. There is no universal hardness value for all tablets, as it varies depending on the formulation and the intended use of the tablet, e.g., immediate release or extended release tablets. However, as a general guideline, normal hardness range, typically, tablet hardness falls between 4 to 10 kilograms. Effervescent tablets, these require much lower hardness to dissolve quickly, usually around 1.5 to 2.5 kgf. Chewable tablets, these are often softer, in the range of 3 to 6 kgf. Test 4. Thickness test. The thickness test is an important quality control measure used in pharmaceutical manufacturing to ensure that tablets are of consistent size. Tablet thickness is closely related to the volume of powder or granules filled into the dye during compression. Consistency in tablet thickness is critical for maintaining uniformity in the packaging process, as well as for ensuring uniform drug dosage and patient acceptance. Principle of the thickness test. Tablet thickness is the physical dimension from one flat side of the tablet to the other, measured in millimeters, mm. During the test, individual tablets are measured using a caliper or a thickness gauge to determine if they meet the specifications set by the manufacturer or the pharmacopoeia. Formula for thickness measurement. The formula for determining tablet thickness in a production batch is straightforward, as it simply involves measuring the thickness of individual tablets and calculating the average, where sigma t is the sum of the thicknesses of the individual tablets, n is the number of tablets measured. Example of thickness test calculation. If a sample of 5 tablets has the following thickness measurements in millimeters: 3.50 millimeters, 3.52 millimeters, 3.48 millimeters, 3.51 millimeters, 3.49 millimeters. To calculate the average thickness, sum of thickness measurements, number of tablets, nn equals 5n, average thickness, 17.50 divided 5 equals 3.50 millimeters. So, the average thickness of the tablets is 3.50 millimeters. Acceptable thickness range. The acceptable range for tablet thickness is typically specified in the product's manufacturing specifications. In general, the thickness tolerance should be within plus or minus 5% of the target value. For example, if the target thickness for a batch of tablets is 3.50 millimeters, the acceptable range might be between 3.33 millimeters and 3.68 millimeters. Test 5. Friability test. The friability test is a crucial quality control measure in the pharmaceutical industry. It is used to determine how easily tablets can break, chip, or crumble when subjected to mechanical stress, such as handling, packaging, and transportation. This test ensures that tablets remain intact and durable throughout their life cycle until they are consumed by the patient. Friability measures the mechanical strength of tablets under simulated conditions of stress to assess how much mass a tablet loses due to abrasion or friction. The tablet friability is calculated by measuring the percentage weight loss of the tablets after subjecting them to a standardized tumbling and rotating action. Principle of the friability test. In this test, tablets are placed in a friabilator, which is a drum that rotates at a fixed speed, typically 25 rpm, for a set period, usually 4 minutes. As the drum rotates, the tablets tumble over and hit the walls of the drum, simulating the type of stresses they may encounter during production, packaging, and shipping. The percentage of weight loss of the tablets due to these stresses is calculated to assess the friability. A friability test is considered acceptable if the weight loss is within a specific limit, generally less than 1% of the initial tablet weight. Formula for friability. The friability of a tablet is calculated using the following formula: friability, % equals, initial tablet weight, final tablet weight, / initial tablet weight, times 100. Procedure for friability testing: sample preparation. Weigh a sample of tablets, typically 10 to 20 tablets, and record the initial weight. Testing in Friabilator. Place the tablets in the Friabilator drum, set the Friabilator to rotate at 25 revolutions per minute for 100 revolutions. This takes about 4 minutes. If the tablets weigh more than 650 mg each, 10 tablets are used. For tablets under 650 mg, a higher number may be tested. Example of friability calculation: suppose a batch of 10 tablets has an initial weight of 6.5 grams before the friability test. After running the test and removing dust from the tablets, the final weight of the tablets is 6.45 grams, initial weight, Y, equals 6,500 milligrams. Final weight, WF, equals 6,450 milligrams. Now, calculate the percentage friability equals 6,500 minus 6,450, divided by 6,500 into 100 equals 0.77%. In this case, the friability is 0.77%, which is within the acceptable limit, less than 1%. Test 6. Disintegration test. The disintegration test is one of the most important quality control tests used in pharmaceutical manufacturing to assess the ability of tablets or capsules to break down into smaller particles when exposed to physiological conditions. This test ensures that a tablet or capsule disintegrates within a specified time, releasing the active pharmaceutical ingredient to be absorbed in the body. Disintegration is a critical step in the dissolution and absorption of a drug, and it directly affects the drug's bioavailability, how well and quickly it enters the bloodstream. Ensuring proper disintegration is essential to guarantee the therapeutic efficacy of the dosage form. Principle of the disintegration test. The disintegration test involves placing tablets or capsules in a liquid medium, usually water or a simulated gastric or intestinal fluid, and subjecting them to gentle agitation. The objective is to check whether the tablets or capsules break down into particles small enough to pass through a standard-sized screen within a prescribed time. This test does not measure how well a drug dissolves but only assesses whether it disintegrates into smaller particles, which is a prerequisite for dissolution. Procedure for the disintegration test. Sample preparation. Typically, 6 tablets or capsules are selected as a representative sample from the batch. Placing the tablets or capsules. Each tablet or capsule is placed in one of the tubes in the basket rack assembly. Immersion in medium. The basket assembly is immersed in a beaker containing the appropriate liquid medium, usually water, simulated gastric fluid, or simulated intestinal fluid, at 37 degrees Celsius plus or minus 0.5 degrees Celsius. Testing. The basket is moved up and down at a controlled speed, 28 to 32 cycles per minute, simulating conditions in the gastrointestinal tract. Observation. The test is run until the tablets or capsules disintegrate completely and no fragments remain on the mesh screen. The tablets should break apart into fine particles within the specified time for the dosage form. Acceptance criteria for disintegration time. Different types of dosage forms have different time limits for disintegration, as per Pharmacopeal guidelines. Some general guidelines are: uncoated tablets should disintegrate within 15 minutes, film-coated tablets typically have a disintegration time of 30 minutes, and enteric-coated tablets should not disintegrate in simulated gastric fluid, pH 1.2, for at least 2 hours but must disintegrate in simulated intestinal fluid, pH 6.8, within 60 minutes. Effervescent tablets should disintegrate within 5 minutes in water. Capsules, hard or soft gelatin should disintegrate within 30 minutes. The specific disintegration time depends on the type of tablet, the formulation, and regulatory requirements. Test 7. Dissolution test. The dissolution test is a key quality control test used in the pharmaceutical industry to evaluate how quickly and efficiently the active pharmaceutical ingredient is released from a solid dosage form, such as tablets or capsules, into a liquid medium. This test simulates the process that occurs in the body, where the drug dissolves in the gastrointestinal fluids before being absorbed into the bloodstream. The rate and extent of drug dissolution are critical factors in determining the bioavailability of the drug, which in turn influences its therapeutic effectiveness. Principle of the dissolution test. The dissolution test involves immersing a solid dosage form, tablet or capsule, in a liquid medium, and continuously stirring the medium under controlled conditions to maintain uniformity. Samples of the medium are taken at specific time intervals to measure the amount of drug dissolved at each point. The goal is to monitor the rate of dissolution, which can be influenced by factors like formulation composition, coating, and manufacturing processes. This test provides insight into the release profile of the drug over time, ensuring that the drug will be available in the body at the required rate to exert its therapeutic effect. Apparatus for dissolution testing. The United States Pharmacopeia specifies two common apparatuses used for dissolution testing. USP Apparatus 1, Basket Method. A rotating basket holds the dosage form, tablet or capsule, in place while it is submerged in the dissolution medium. The basket is rotated at a specified speed, typically 50 to 100 revolutions per minute. USP Apparatus 2, Paddle Method. A paddle is used to stir the dissolution medium while the dosage form rests at the bottom of the dissolution vessel. The paddle also rotates at a specified speed, usually 50 to 100 rpm. This is the most commonly used method. Other apparatuses, for specialized dosage forms. USP Apparatus 3, Reciprocating Cylinder, used for extended release formulations. USP Apparatus 4, Flow-through cell, used for poorly soluble drugs or modified release dosage forms. Dissolution medium and conditions. The dissolution medium typically consists of simulated gastric fluid, pH 1.2, or simulated intestinal fluid, pH 6.8, to mimic conditions in the stomach or intestines, respectively. Water and buffers may also be used. The volume of the medium is usually 500 milliliters to 1000 milliliters. The temperature is maintained at 37 degrees Celsius, reflecting body temperature. Procedure for dissolution testing. Sample preparation. Select a representative sample of 6 to 12 tablets or capsules from the batch. Testing setup. The dosage form is placed in the basket or paddle of the dissolution apparatus, and the dissolution medium is maintained at 37 degrees Celsius. Stirring. The basket or paddle is rotated at a specified speed, usually 50 to 100 rpm, ensuring that the dosage form is exposed to the medium under controlled agitation. Sampling. Samples of the dissolution medium are taken at specific intervals, e.g., 5, 10, 20, 30, 60 minutes, to measure the amount of drug dissolved at each time point. Analysis. The concentration of the dissolved drug in each sample is measured using a suitable analytical technique, such as UV visible spectroscopy or HPLC. Calculate percentage dissolved. The amount of drug dissolved is expressed as a percentage of the labeled drug content. This is plotted against time to generate a dissolution profile. Acceptance criteria for dissolution. Pharmacopoeias set specific dissolution criteria for different dosage forms. Typically, a dissolution specification is set as a percentage of the drug dissolved within a certain time frame. For example, for immediate release tablets, a common specification is that 80% of the labeled drug amount must dissolve within 30 minutes. Extended release or delayed release formulations may have different time specifications, depending on the desired release profile. The acceptance criteria vary by drug and dosage form and must comply with regulatory guidelines. Test 7. Assay testing. Assay testing is a critical analytical procedure used in the pharmaceutical industry to determine the content or potency of the active pharmaceutical ingredient in a tablet or other dosage forms. It is one of the most important quality control tests, ensuring that the drug product contains the appropriate amount of the active ingredient as specified in its formulation. The accuracy and precision of assay testing are essential for confirming that each batch of the drug meets regulatory requirements and delivers the expected therapeutic effect. Principle of assay testing. the principle of assay testing is to quantitatively determine the amount of the active ingredient in the drug product using analytical techniques. The most common techniques for assay testing include high-performance liquid chromatography, widely used for separating, identifying, and quantifying the active ingredient in complex formulations. UV visible spectrophotometry, used to measure the absorbance of the active ingredient in a solution, which is directly related to its concentration. Titration, a classical method where a solution of known concentration is added to react with the active ingredient until the endpoint is reached. Gas chromatography, used mainly for volatile compounds or APIs that require specific separation techniques. Mass spectrometry, used in conjunction with other techniques to provide precise and accurate quantification of APIs, especially in complex mixtures. Procedure for assay testing. The general procedure for assay testing involves several key steps: sample preparation, weighing. A specific number of tablets, typically 10 to 20, are weighed, and an average weight is calculated. Dissolution or extraction. The tablets are dissolved in a suitable solvent to extract the active ingredient for testing. Dilution. The solution is diluted to a suitable concentration that can be measured accurately by the chosen analytical technique. Analysis. The prepared solution is analyzed using the selected analytical method. For example, if using HPLC, the solution is injected into the chromatograph, and the active ingredient is separated from other components. The detector measures the concentration of the API based on the area under the peak. Calculation of API content. The result is compared to a standard or reference substance to determine the amount of active ingredient present in the tablet. The formula used for calculating the assay of the active ingredient is as follows. Acceptance criteria for assay testing. Pharmacopeal standards typically specify the acceptable range for the content of the active ingredient in a tablet. The acceptance criteria for most drug products are as follows: typically 90% to 110% of the labeled claim for the active ingredient. For potent drugs, low-dosage drugs, stricter limits may apply, such as 95% to 105% of the labeled content. These limits ensure that the drug product delivers the correct therapeutic dose and remains safe and effective. Thanks for watching. If you found this video helpful, be sure to like, comment, and subscribe for more videos.